Canada. His PhD advisor was Dr. Hilary Maddin, who some of you may remember from PaleoFest in Rockford last March, which was pretty much the last public event before the COVID shutdown. Arjun co-authored articles describing 2 new species of microsaurs from Mazon Creek in 2019. He's currently transitioning to a postdoc position at Harvard and doing his work remotely. I had hoped to have him speak to us in person last year. That didn't work out, but we wanted him to have a chance to visit and collect at Mazon. So I've given him an open invitation to visit here during the warm months post-COVID, and we'll find some way to get him into the sites to collect. And tonight he's going to tell us about new discoveries from Mazon Creek. Arjun.
All right, I'm just going to share my screen. Well, hi everyone. My name is Arjun Mann, for those of you who don't know me. I'd like to thank everyone first for inviting me to give this talk today. I've been following ESCONI's exploits for some time. Since I started as an undergrad, really, your website has been a great resource for me. And it's great to give you this talk today, albeit virtually. Today, I'll provide you with a lecture on my doctoral research that I recently conducted at Carleton University over the past 4.5 years. And my research mainly concerns the systematics and ecomorphology of amniotes across the Carboniferous, and specifically at Mazon Creek, Illinois. So with any more preamble, let's get to it. All right, some introductions. Just so everyone knows what I'm talking about, what is an amniote anyways? In a nutshell, they are vertebrates that have an amniotic membrane, sometimes called a cleidoic egg. They include all living and extinct reptiles and synapsids, the latter being the group that includes mammals as well. So you and I are both amniotes. The advent of the amniotic membrane marks the permanent colonization of the terrestrial realm by tetrapods. And tetrapods are the clade that include all 4-limbed vertebrates. And here's some images on the slide for you. Now, amniotes originated sometime during the Carboniferous, possibly as early as the Mississippi and Pennsylvanian boundary. This event is marked by the permanent colonization of land that was attempted previously by amphibians. The oldest fossil amniotes are about 318 million years old, and they're from the deposits of Joggins, Nova Scotia, where the earliest reptile, Hylonomus, and the earliest synapsid, Acifestra, are both found in lycopsid tree stumps. In general, amniote fossils are exceedingly rare in the Carboniferous, as some of you may know. They only show up in great diversity towards the Permocarboniferous boundary. And this later diversity is possibly aided by climate change causing the breakup of the large Carboniferous swampland environments into a variety of more arid dryland environments. And this series of arid dryland environments would have accommodated a pre-adapted amniote community and leading to their evolutionary success. This being said, the lag in the timing of diversification of amniotes from when they first arose to their peak Carboniferous diversity, marked by the 2 red lines on this figure, is somewhat odd for what theoretically should be qualified as an adaptive radiation or rapid radiation that is usually characterized by an increase in ecological opportunity in the terrestrial realm. It's possible that this lag is due to a sampling error or bias in the fossil record, a question that my thesis aimed in part to address. And to the last point, a recent hypothesis has arisen again in the literature debates of amniote origins that perhaps a small group of reptile-like tetrapods known as microsaurs are in fact true amniotes. This particular new version of the hypothesis put forward by Pardo et al. 2017 places the fossorially adapted recumbirostrans microsaurs, so adapted for digging, within early reptiles. This inclusion would increase the diversity of amniotes at their onset from 1 to several genera. Studies on the early record of this group would alleviate details of the origin of amniotes, including their true ecological and taxonomic diversity. Several new records of recumbirostrans fossils have been found from the Carboniferous of Mazon Creek, which is the locality that I'll introduce briefly today to you guys, although I know that this group's probably well informed all about it. In general, the Carboniferous landscape was defined by these large overarching swampland environments with tall mangrove-like forests and large insects everywhere. Some images from artists of reconstructing this environment. And the Mazon Creek locality specifically was at the terminal ends of this swamp system as a river delta. Mazon Creek is a collection of strip mine localities of exposed Francis Creek shale strata that are aged by the Colchester Coal No. 2 seam to approximately 307 to 309 million years old by the latest studies. These sites are considered a lagerstätte, or area of exceptional soft tissue preservation. Paleo-environmentally, the system is an estuary and marine-influenced river delta, analogous to the modern Mississippi Delta, perhaps. And you can see in this slide, you have the location of Mazon Creek within modern-day Illinois, the location of Illinois within Pangea during the Carboniferous, and at the bottom, there's a map of the various pits that make up the Mazon Creek lagerstätte. And there's also a little picture of an active locality on the Mazon River. Mazon Creek is Muscovian or Westphalian sea in age, so slightly younger than classic Carboniferous localities such as Joggins, but older than sites such as Linton and Nizhny in the Czech Republic and Linton in Ohio. There's another outcrop photo for you as well, probably taken right from the ESCONI website. Some of you might remember this or know where this locality is. I hope to get there one day. And this is a graphic of the world during the Carboniferous late Pennsylvanian, showing the principal Westphalian tetrapod localities across the supercontinent of Pangea. Note the very distal position of Mazon Creek near an inland sea, which usually accounts for its high number of marine organisms. Now, Mazon Creek is obviously notable for its unique preservation of organisms and siderite concretions that can be split in half to reveal both parts and counterparts of animals. The fossils are often preserved with soft tissue such as body impressions, and Mazon Creek can be divided into currently washout and in situ marine fauna. These terms have kind of replaced the old Braidwood and Essex motifs, although I'm sure they're probably still used. Tetrapods come from nearshore terrestrial erratics. They're not in situ members of the fauna. That does mean they were probably inhabiting very distal ecosystems. These tetrapod fossils are usually preserved as natural molds that are infilled with kaolinite, among other minerals, and can be studied using latex peels or modern technology such as CT scanning. And this is just a snapshot of the diversity of Mazon Creek organisms, including everything from plants, worms, tetrapods, to a few specimens of amphibians and Phlegethontia. Now, Mazon Creek has had a long history of collection and research at the site by researchers such as Cope and Newberry and Roy L. Moody. However, the site has proven much less productive for tetrapod research than sites such as Linton, Ohio. Roy L. Moody said every 1 in 100,000 concretions is likely to produce a tetrapod. I know some people have inflated this to 1 in a million, but nevertheless, Mazon Creek remains a very difficult locality to produce a lot of tetrapod research at, or at least so that was thought. And despite this, it does bear some of the oldest members of really important lineages, including Amphibians grandiceps, the oldest stemless amphibian currently. A little more recently, research at Mazon Creek concerning vertebrates includes studies by Baird, Bardak, and Zangirl on the Paleozoic fish. Joseph T. Gregory and Bob Carroll provided major studies on Cephalurpaton venturamatum, the only reptile known from Mazon Creek. And Dr. John Balt provided a comprehensive study on amphibians, setting the stage for modern list amphibian research. In this figure here, these are SEM plates showing bicuspid pedicellate teeth in amphibians, which is a trait that's used to link amphibians to modern amphibians. Finally, Drs. Balt and Baird cultivated valuable relationships with the private fossil collecting community and consolidated a lot of new tetrapod material at the Field Museum and museums in the area. And it's because of their work in this that I was allowed to do my thesis, because a lot of the collections that I used for study was due to their efforts. So thanks to them for that. And recently there has been a resurgence in study at Mazon Creek concerning largely the invertebrates in geological setting, but also some really nice studies on the telemonster. And these are pretty high profile papers, but nothing really on the tetrapod fauna. So that's where I came in. And just to, not to do anyone any injustice, but the last real major study of the Mazon Creek vertebrates was done by Stephen Godfrey in 1997, who published a few papers, but also published the most accepted tetrapod fauna list from the site, showing relatively low diversity, 24 concretions, and only one amniote known from the site. So right now, we have a lot more data, so hopefully we'll change this a bit. And with this new data, this thesis that I did aimed to address three primary questions. First, what is the alpha taxonomic diversity of the amniote fauna at Mazon Creek? And second, what is the ecomorphological diversity of the amniote fauna at Mazon Creek? And third, can this fauna tell us anything about the early radiation of amniotes? And to do this, these questions were addressed through a series of systematic studies describing 4 new recumbirostrans taxa and redescribing historic amniote remains such as Cephalorpaton. This was done using comparative anatomical analyses based on modern imaging techniques, including CT scanning, scanning electron microscopy at the Canadian Museum of Nature, using a Geol SCM machine that is pictured at the bottom right. I also implemented Bayesian and parsimony phylogenetic methodologies to build a tetrapod tree of life for these guys, using data from the recent study of Pardo et al. 2017. In total, the comparative data set was gathered from over 14 academic institutions across both Europe and North America, which is how I spent the first 2 years of my dissertation, basically traveling to every major institution to make sure that I could see every major tetrapod specimen and get a good handle on early tetrapod anatomy. And the results of this research first led to the naming of the new genus Diablorodor balti, named in honor of the late John Bolt, who unfortunately passed away before this study was published, although I did have the opportunity to meet with John several times, and he helped me at getting started on this project quite a bit. Diablorodor, just to go over the anatomy quickly, has a short and wide snout, wide skull rather, with a co-ossified brain case and occipital region. This region is sometimes called an os basal or auto-occipital. There are 23 small homodont teeth within there, a thin maxilla, approximately 20 dentary teeth, and the skull is ornamented with radiating grooves and large pits on the frontal and prefrontal. This also bears a unique single-piece palpebral ossification, which is an eyelid bone ossification, which can be seen right here. Postcranially, Diablorodor is an extremely short-bodied animal with only 17 presacral vertebrae. The limbs and girdles are robust, and overall the body length is slightly rotund, as you can see through the expansion of the rib cage. Phylogenetic analysis of this new animal, Diablorodor, under both parsimony and Bayesian frameworks provides the same result as an early diverging brachystelicid that is sister taxon to Permian-aged members such as Corolla and Batrapedes. In addition, all the recumbirostrans is recovered within amniota in this analysis, as in the analysis in Pardo et al. 2017, meaning that these are basal reptiles. The addition of Diablorodor to the Brachystelicida permits a reassessment of the anatomy within the clade as well, including characterizing the group as having both short and wide co-ossified skulls with a reduced postorbital region and a single piece eyelid ossification. Here's just a comparative diagram of all the known brachystelichids and their skulls. They're very short, squat things, as you can see. Postcranially, brachystelichids are all short, wide-bodied animals with well-ossified limbs and pectoral girdles. As you can see here, they are quite wide-bodied, some of the animals here. And this is one of the only recumbirostrans groups to actually show amniote-like gastral ribs, which can be seen here sneaking through the rib cage. That is one of the features which links them to actual reptiles. The co-ossified crania of brachycoelichids and the robust postcrania indicate that brachycoelichids were likely terrestrial burrowers of some kind, using their limbs and girdles possibly to push dirt and soft soil out of the way. So they're possibly more limb-oriented burrowers rather than head-first burrowers. Also, derived Permian brachycoelichids such as Corollambatropedes show unique tricuspid teeth that are shared with modern aquatically herbivorous reptiles such as the marine iguana and Blorhynchus cristatus. It's possible that this tricuspid dentition were used to rake algae in a similar manner. This would explain their presence at a very aquatic locality such as Mazon Creek. An anterior-facing dentition might suggest that they were used to actually rake algae in a similar manner. And now our next chapter deals with another new species I named called Infernovenator stene, whose anatomy provides some clarification on the origin of the snake-like group of recumbirostrans known as Lysirophians, or Molgophidae by the formal name. The cranial anatomy of Infernovenator reveals a more stereotypically recumbirostrans skull than previously known Lysirophians. Before, Lysirophia was included in its own order. Now it is deeply nested within recumbirostrans microsaurs. This includes, in this animal, our new animal, Infernovenator, a well-developed circumorbital series. That is the bones that line the eye hole, and a well-developed snout region. Additionally, Infernovenator is a shovel-snouted ecomorph, referring to the shovel-shaped morphology of the snout that's used for digging that it shares with other recumbirostrans taxa, such as Alotrimity gasculae, pictured in the CT data on the bottom left. Postcranially, all Lysirophians, like all Lysirophians, Infernovenator has a long snake-like body with a unique 61 presacral vertebrae. So it's a really long guy. Unique here also, the hind limbs appear particularly well developed with a large femur, but the forelimb is significantly reduced, perhaps only to 2 humeral elements. This will definitely become more important in a review of limb reduction in chapter 6 of my thesis, but it's a very odd looking guy. It's got no front limbs basically. And our parsimony and Bayesian phylogenetic analysis of this guy recovered it as sister taxon to Brachydectes newberrii, one of the best known Lysirophians in the Permo-Carboniferous. And that's not surprising because there's not many valid taxa of these guys around. But more importantly, we're able to use this phylogenetic analysis to identify some aspects of skull evolution in fossorially adapted recumbirostrans, including mosaic evolution of the snout and cheek modules. We are able to show here that cheek emargination and snout shape evolved independently multiple times convergently within recumbirostrans. Additionally, recumbirostrans houses a variety of snout ecomorphotypes present, including round-headed, shovel-headed, and spade-headed ecomorphotypes that are analogous to ecomorphologies that are observed in modern fossorial or burrowing lizards, such as amphibians. And there's one of them pictured at the bottom here for you. In this figure, we can see that the pink boxes show cheek emargination and shovel-snouted ecomorphs evolving independently at least twice within the group, both in a stotolepid, such as Paleozoic, and within our new guy, Infernovenator. So that's convergent evolution at work for you guys. Finally, in this chapter, we show that Lysirophians are the most diverse speciose clade of recumbirostrans tetrapods in Mazon Creek, at least represented by 3 taxa. These are Infernovenator, the new guy, Brachydectes, and another new taxon that we'll discuss briefly in chapter 6. So we'll get to that later. And this taxon chapter 6, one of the specimens was actually found by Eugene Richardson, and that's pictured in panels E to F here. And we'll go over that in a bit later. Oh, next, we review historic amniote remains from Mazon Creek, and this includes the basal reptile known as Cephalorpaton venturamadum. Quick note about the fossil here. So this is the actual fossil of Cephalorpaton, but the cast beside it, panel B here, is a resin cast that was actually taken shortly after the animal was actually found. So it's quite old, and I was able to relocate this at the Yale Peabody Museum when I was there. The anatomy of Cephalorpaton betrays a very thin and gracile animal with a lightly built skull and slender arms, similar to later diapsid reptiles. There are defining 16 large conical teeth in this animal in the maxilla, significantly enlarged maxillary teeth compared to their dentary counterparts. And overall, the anatomical analysis supports interpretations that Cephalorpaton as a basal reptile, but with close affinities to diapsid reptiles. So that's quite advanced for its stage in the Carboniferous already. And one of the most important and surprising discoveries to come out of this study, however, were from the referred remains of Cephalorpaton at the slightly younger canicle site called Linton, Ohio. Cephalorpaton prior was one of the only basal reptiles identified from multiple Carboniferous localities. But here we were able to show that these remains from Linton bear resemblance to another group of early reptiles called parareptiles, specifically the ecleistorinid parareptiles. Parareptilia has classically been regarded as the sister group to eureptiles, a completely extinct clade, which some people have argued have given rise to turtles, although that's hotly debated. But recent studies have suggested that parareptiles may deeply nest within diapsids, which point to an earlier radiation of the entire diapsid clade within the Carboniferous. And before this study, the oldest parareptile was named by Modesto and colleagues from the terminal Carboniferous of PEI Canada, that's Prince Edward Island, and is the beautifully preserved Erptonix arsenaltum pictured here. It's quite a lovely animal. We named the new oldest parareptile Carbonodraco lundi, which is basically translates to Dick Lund's coal dragon. Dick Lund was the founder of this animal. I don't know if any of you guys are familiar with his work, but he is a emeritus curator at the Carnegie Museum currently. And this animal bears a short snout where the high facial lamina of the maxilla cuts the lacrimal out from the external nares. That is the main difference between this and a Cephalorpaton. This quite different configuration of the snout and the fact that this animal shares a large premaxillary fang with later ecleistorinid reptiles such as Colobomicter from the Dolise quarry really show that this animal is a different taxon. And for comparison, you can see how Carbonodraco fits well within the motif of other ecleistorinid parareptiles pictured here. This group is defined by their short snouts and unique pitted ornamentation. You can see the similarities between the long fang and Colobomicter, reconstructed up top and pictured in a few different panels, and Carbonodraco's fang-like premaxillary dentition, which reaches approximately the level of the two maxillary canines. Now, the record of ecleisterinidae and most parareptilia has been restricted to the Permian for some time now, with most models already noting major ghost lineages in the evolution of this clade. However, Carbonodraco occupying a relatively derived position as an ecleistorinid would push the diversification of parareptiles even earlier than expected, with a radiation of parareptiles probably happening during the Muscovian, a little earlier than Mazon Creek. For your eyes, here's a scientific reconstruction that was done by my paleoartist friend, Henry Sharp. And I'll leave that up here for a second for you all to enjoy. Alrighty, so now we get to the stuff that's in the process of being published. All these papers are currently either in review or awaiting review and are submitted. So in this section, we introduce the world to another new genus. I call it Jormungand balti, named in honor of John Balt. And we review the integumentary evolution within Recumbirostrans based on this exceptionally preserved new animal. A quick side note about the name: Jormungand is the name of the Midgard serpent in Norse mythology that Thor does battle with at Ragnarok. I think that's a cool and unique name for a serpentine-like animal, and we'll give the animal a little bit more media coverage, hopefully. Jormungand's skull is round with a co-ossified occipital and braincase region, albeit with a distinct supraoccipital element. The snout is short with anteriorly located orbits, and there's reduced tooth row filled with 14 small peg-like maxillary and dentary teeth. Postcranially, anatomy of Jormungand reveals a long-bodied morph with approximately 40 presacral vertebrae, so a little less than a foraminator, and at least 17 caudals. So a bit of a short tail. The limbs appear long but incompletely ossified, so this may be a little bit of a juvenile. And the soft tissue impression shows a interesting tubular-like body plan, similar to modern fossorial reptiles. Parsimony phylogenetic analysis of Jormungand reveals the taxon is sister to all brachystelichids within the clade Coxytenoidea, to the exclusion of Lysirophians. That's a little technical, but basically it sits between Infernovenator and Diablorodor for its relationship. But most importantly about this new animal, there is preserved a full suite of dorsal and ventral dermal scales. These circular scales are overlapping in morphology all over the body. The SEM images show these scales reveal superficial ultrastructural ornamentation that comprise both of longitudinal ridges and pits. These bear similarity in structures that are observed in modern fossorial reptiles. Here are progressively closer scanning electron micrographs of the scales, showing longitudinal ridges, as I mentioned before, and giving an overall shell-like appearance to the scale. Pits are usually singular in between the ridges, but can be irregularly paired. So there's a bit of irregularity to this. And a schematic of a single scale is seen in panel D, just to simplify these things. There is some cranial scale differentiation within Jormungand. The SEMs of the cranial region show no ridges on the scales, indicating that there were at least smoother in this region. Similar cranial scale differentiation is often seen in extant reptiles. Here's a schematic of cranial scales in a modern living skink that shows the type of differentiation that might have been present in this animal. There is some possibility that the scales we're observing are in fact keratinous, or at least partly keratinous in origin. This includes the ornamentation. This is in contrast to dermal scales or osteoderms composed of bone, which are normally preserved at Mazon Creek as impressions in Mazon Creek tetrapods. But conversely, these scales seem to be preserved in the soft tissue impression and can only be picked up using methods such as a scanning electron microscope. It is possible that these may be the first record of soft tissue scales for an animal of this kind, but a little more work is needed to prove that. So right now, we're just going to retain these dermal scales made of bone. In extant reptile scales, ultrastructural patterns have both functional and ecomorphological significance. The ultrastructural pattern of the scales of Jormungand bear resemblance to fossorial reptile scales that are usually adapted for shedding dirt. These include mesh-like repeating patterns that are linearly arranged. And on the figure, we can see the variety of patterns that are found in snakes. But I did some images of my own at the Canadian Museum of Nature using an SEM. Just to hone the ecological nature of these scales. This is the scales of a basilisk lizard, or Jesus lizard as it's sometimes called, which is a terrestrial climber and poorly adapted for shedding dirt because it doesn't really interact directly with the soil. You can see here in this animal, there are actually dirt crystals wedged inside the holes, the lattice of this scale, which are all that shiny material in between. But conversely, if we look at an SEM of a fossorially adapted skink with a clean surface, you see here these linear arranged patterns which don't let dirt get wedged in there. So that's sort of the thing that we're seeing in Jormungand, and we're interpreting this as an adaptation for fossoriality. Finally, based on the new data from Jormungand, we were able to summarize integumentary evolution within Recumbirostrans, noting the loss of osteoderms and other dermal ossifications, including gastral ribs, which can be associated with body elongation in the group and likely increased fossoriality and burrowing within the group. This reduction is possibly due to functional constraints associated with locomotion, such as sidewinding. If you need to move and undulate through soft waters or even land, you probably want to reduce the amount of bone weight you're carrying. Nearly all members of Recumbirostrans retain some form of thin, ornamented dermal scales, similar to these observed in Jormungand. But a variety of them are lacking these dermal ossifications. Finally, and the last new animal I'm going to show you today is this, we're kind of toying with the name of Farewell to Arms for the paper. I'm a big fan of the Ernest Hemingway book, but we're calling this Nagini, named after a snake, Hemingwayi, which represents possibly the earliest example of amniote-like limb loss in a Paleozoic tetrapod. This Lysirophian basically completely lacks its pectoral apparatus and forelimb, but retains a hind limb. So here we can see both the adult and juvenile referred material of this animal. The juvenile specimen was found by and prepared by Eugene Richardson at the Field Museum, and the adult animal was pointed out to me by David Duck, a paleo artist in your group, who found it at the Milwaukee Museum and let me know about it. So as you can see here, there's no elements of the pectoral girdle or forelimb in A and B, the first animal here. The second one does bear a well-developed hind limb, but also lacks completely the pectoral and forelimb, which is a very unique phenomenon, similar to Infernovenator, but Infernovenator, there was something, possibly a rudimentary forelimb present. This guy's got none at all. It also has a higher presacrovertebral count, unlike Infernovenator, which had 61. Here we have around 82 to 85 presacrovertebrae present. So this is a very snake-like long animal. The referred juvenile material is the smallest documented Lysirophian known and shows incomplete ossification of the skeleton. Scanning electron micrographs reveal the specimen is comprised mostly of pyrite rhamboids, not kaolinite. And this indicates the preferential preservation of pyrite, that this animal is probably mostly soft tissue and barely ossified upon fossilization, and is likely neonatal. And these are progressively increased resolution scanning electron micrographs and showing the pyrite rhamboids in panel D. Parsimony phylogenetic analysis of this new taxon recovers the species sister to Infernovenator to the exclusion of Brachydectes. So it is a closely related Lysirophian to Infernovenator. Additionally, Recumbirostrans is recovered with an amniota, as has been the case throughout this thesis, as reptiles. This phylogenetic analysis also allows us to do some comparative phylogenetic work to compare limb reduction and body elongation over the entire Recumbirostrans. In extant fossorial reptiles, the two are usually correlated, and this appears to be the case within Recumbirostrans. On the time-calibrated phylogeny here, we can see that the darkest silhouettes indicate body elongation at its highest, and limb reduction is indicated progressively by orange, pink, and blue dots. Blue being the complete loss of a limb module. So we can see in Nagini and Infernovenator, we have the complete loss with a really elongated body form. So these two modes are working in conjunction. And the antiquity, how old this is appearing within the fossil record of both amniotes as well as Recumbirostrans, reveals this may have been a prominent adaptive mode for amniotes across their entire evolutionary history. One of the ways that reptiles and amniotes diversify in the fossil record, becoming fossorial and changing their body plan. And now Nagini also permits some speculation into the multiple developmental pathways to limb reduction, including both limb reduction and loss within Recumbirostrans. In general, limb development can be broken into three phases. An initial patterning phase where a limb bud in the embryo is specified, and a second, a limb morphogenesis and patterning phase where a limb bud grows and forms, and third, longitudinal outgrowth phase once the animal is actually growing the limb in length. And disruptions to any of these phases in development leads to a unique outcome of the animal in life. So failure to specify a limb bud field in the genetic component means you lose the entire limb. Disruptions to having a limb bud means you sometimes lose distal limb modules. And disruptions to the third phase, the closure of the growth plate, means you get shortened limbs. And what appears at Mazon Creek, comparatively across these animals known, is you have all three of these modes of limb reduction acting in one fauna, with Cephalopiton with a really well-developed limb being the basilar eureptile outgroup comparator. Then you have Diablorodor compared to it a shortened limb form, a form like Jormungand where you're missing distal elements, same with Infernovenator. And then finally, with an animal like Nagini, where you have no limb module or pectoral girdle and a failure to form those elements developmentally to begin with. And this is all in one fauna. The complete loss of the pectoral and forelimb in Nagini indicates that there was a failure to establish this limb field in this taxon. If you recall, that's mode 1. This limb reduction mechanism has been identified in snakes and caecilians as the primary limb loss mechanism and involves the homogeneous expression of a limb gene known as TBX5 over the whole flank. This mode present in such an old form such as Nagini means that this is the earliest evidence of amniote-like limb loss, like a snake in the fossil record, which is a really interesting, unique discovery. So the conclusions of the study, we made it here. So over the course of this thesis of mine, we were able to reassess the alpha taxonomic diversity of Mazon Creek amniotes particularly, revealing several new ecologically diverse species, showcasing a range of adaptations for fossoriality and terrestrial ecologies, with their unique body forms and limb morphologies highlighted. Additionally, this thesis provides the first comprehensive reassessment of a recumbirostrans fauna to begin with, showing that this group firmly was rooted in the Carboniferous, ultimately stressing that this is an older adaptive radiation of amniota, and there's a lot more terrestrial reptilian components to the Mazon Creek fauna than previously thought. And I was also able to revise, tentatively, the systematics of the entire Mazon Creek tetrapod fauna, based on my travels and all the specimens that I've seen, showing really that this is a diverse fauna on par with Linton and other Carboniferous localities, just the specimens haven't been studied correctly. And this is interesting compared to what other previous authors have noted, that Mazon Creek might have just been a depauperate fauna, and that's really contrary to this. Some new additions to this list is I actually found a specimen of Carbonodraco lundi from Linton at Mazon Creek, and it's this animal up here pictured, Amphibians thoracatus. It's a little difficult to see, but we'll be publishing this in the next coming year, and is essentially the same animal and a second big reptilian from the site. And that leads me to my future directions. There's a hell of a lot more fossils turning up from Mazon Creek, and I intend to keep working on this fauna, particularly now moving to revising the anatomy of the historic amphibian component. This is a picture of the lost type of amphibians that Cope described. And we've already made some headway with a paper coming out earlier this year in JVP showing the earliest evidence of list amphibian-like toe pads in amphibians. But we've got a lot more work to do revising the anatomy of this animal. If anyone knows of new specimens of this animal, I'd love to see them. Please get in contact with me. It'll be a great project. I'm happy to involve people in the research process. I'm also starting work more broadly on tetrapod Carboniferous localities, including Linton and 5 Points, Ohio. This is a new animal from 5 Points we're working on, preserved in coal, so completely different stuff. But that's another story, and I'm happy to come give another talk on that one day as well. And just before we get to questions, I'd like to end off by sincerely thanking everyone that helped me out along the way, my family, friends, academic colleagues, and everyone who came to this talk today, of course. Thanks for listening. I must say a lot of this research was allowed by the cooperation of the paleontological community. such as yourselves. I had a lot of help from one of your members, paleo artist David Duck, who helped me locate some of the specimens in this study. And I'm just putting a general call out to you for further help. We, as paleontologists, always need the help of citizen scientists and researchers. If there's more tetrapod material out there, please send me pictures and everything of it, and I'd love to see all the specimens. All right, I think that's it for me.
Can we clap now? Nice job, man. Really nice. Well done. Brilliant.
All right, am I still sharing my screen?
Sure.
Yes.
Oh, I think you stopped sharing.
Oh,
So we're
perfect.
seeing
Okay.
a picture.
Yeah, please let me know if you'd like to see anything again, too. I don't know how fast I was going.
Arjun, are there any tooth morphologies that are perfectly unique to the tetrapods or any group within the tetrapods?
Perfectly unique. Tetrapod teeth are different than fish teeth in a lot of ways. They bear different tissues, but this sort of cuspate morphology that you can get in things like brachystelichids, I showed that tricuspate dentition. That's quite unique to this type of tetrapod group. Also Cephalorpaton, it's massive. If you look at the animal, it's really got these massive cone-like teeth. That is very unique among tetrapods of its age. So clearly within this group, they were experimenting with a variety of diets, not just eating the same thing, and really compartmentalizing what they did.
I noticed in, I think it's Colobomicter, is that right?
Yeah, yeah.
I saw those ridges at the base of the teeth that were pictured. Is that a unique trait that would only show up in Colobomicter, or because I do a lot of digesting of material in acid, and so I come out with all these loose teeth, and for the most part I'm looking at fish teeth or sharks or whatever, but every so often something comes up that's, you know, a little different, like that claw that I sent to you, and
Yes.
I don't quite know what to do with it.
Yeah, so with the teeth, the little ridges you're seeing are called enamel fluting. They're actually kind of crenulations in the enamel. And towards the base, you have folding in the tooth, and that's called plissadentine. They are very common in amniotes. When you get to amphibians, you have a condition called labyrinthine infolding, which you get sort of like you have to see it broken and then you see a convoluted maze of dentine. But within amniotes, that sort of light fluting on the teeth, that is pretty diagnostic to say it's an amniote, but not very diagnostic to say what type of amniote it is because it's shared within multiple groups.
Okay. Yeah. Okay.
Do you have any idea which localities are more likely to yield things like the amphibians and the reptiles?
You know, a lot of the, I didn't go over the locality data from where these found, but a lot of the tetrapods do come from what is called pit 11.
Okay.
I don't know if that's because there's more tetrapods there. My suspicion is because that's more sampled. It's sort of a sampling bias. Because more people have collected from pit 11, you're more likely to find a tetrapod just by sheer number. Within Mazon Creek, I haven't heard of a place yet that bears a more terrestrial fauna by the pure numbers.
Because that's more marine and generally thought of more wash-in, you know, where the plants are found or where, you know, maybe some of these tetrapods would have lived, although I guess we don't really know.
Yeah, I think that in general, the tetrapods are probably erratics that are washed out, but I think it just comes down to there may actually be a more terrestrial part within Mazon Creek, but no one's put the numbers to the game and actually done tabulations of that to account for how hard each locality is being sampled. And right now, pit 11 is just being sampled way more than any other place. So within museum collections, you have entire collections. The Royal Ontario Museum has one by Kathleen Ramsdale. I don't know if the name It was donated sometime in the 90s, but that entire collection is PIT 11. And it's about a good chunk of 500,000 concretions sitting there. So it's a big collection. And there are they have a few aquatic amphibian taxa that I don't know if are new, but they are fossils that I looked at in my dissertation.
We still visit Pit 11.
Yeah.
It's unfortunately fairly overgrown now, but in the spring, up until maybe early June at the latest, when the ticks come out, you could still go and still collect concretions there.
Yeah, I'd love to go. If you guys want to take me when I can get stateside, I think that'd be a hell of a time.
We would take you, definitely.
Perfect.
Did you consult with Charlie Shabika on any of this material? I don't know if they had. I think Northeastern still has some of the concretions. I'm not sure what they're doing with them, but Shabika at one time was big into the Mazon Creek material.
Yeah, so I actually my second year of grad school cold called Charlie Shabika. I took the phone up, grabbed started looking for his number, and
old Charlie.
Yeah, and I picked him up and I just said, hi, I'm a grad student studying Mazon Creek tetrapods. Do you have any? And he told me a whole shebang. He had donated most of his tetrapod material to John Balt at the Field Museum by that point. But from what I understand, he told me he sold part of the counterparts to a man from Japan. And then I asked, oh my God, I need those counterparts. So if you look at that pretty amphibian specimen I showed you, that's actually in the Mazon Creek fauna book with the body outline, the other half of that thing is somewhere in Japan. So I got the name of the agent, who is Roichi Ibasawa. I remember that name. And I tried to search for him. I contacted Japanese paleontological colleagues and turned up nothing after a year of searching. So I think the next step is at some point to get over to Japan and start searching the museum systems myself. But yeah, half them are in Japan.
Charlie.
Yeah, but he was very helpful.
Oh yeah. One other question. I couldn't see the cladograms that well. What's the relationship between the Mazon Creek tetrapods and the Acanthostega, Ichthyostega group?
Yeah, so compared to Acanthostega and Ichthyostega, which are considered more fish-like stem tetrapods, we're nesting these Mazon Creek tetrapods within reptiles. So quite a bit more derived. So within Amniota, you have the sister groups Synapsida, which are us, and then you have reptiles. These are nesting within the earliest reptiles. So not
they'd be more of the stem group of the reptiles.
Yeah, around there. Yes.
Yeah.
Is the term microsaur kind of an informal kind of basket term for elevating the amphibians up to kind of a reptile status?
Yeah, so microsaur is kind of the opposite. So microsaur is an informal wastebasket term, correct? But it's actually degrading them towards amphibian status.
Okay.
These animals are actually probably more closely related to reptiles, whether they're crown group reptiles or just slightly on the stem. They certainly share more traits with reptiles than they do with amphibians. So for instance, at the site, the two polarizing factors are you have Cephalorpiton, a clear reptile, then you have Amphibamus, a clear amphibian. All these new specimens that I've found and have named bear more resemblance to Cephalorpiton than they do Amphibamus. Yeah.
One other question. There was a legless amphibian that was figured in our club's fauna book. I don't know the pronunciation. Phlegethonicia.
Phlegethontia.
Yeah. Is that distinguished from what you had identified from the Milwaukee Public Museum?
Yeah. So when I got the specimen ID from David and the curator, they called it Phlegethontia. And I looked at it and looked at the vertebrae, particularly the vertebrae between Phlegethontia and Aeistopods is the group name, and these microsaurs and reptiles are quite different. So that was one of the ways I was able to identify it, and it is quite distantly related. Amphibians like Phlegethontia are actually more closely related to animals such as Ichthyostega and Acanthostega. they're actually the oldest fish-like tetrapods to reverse the limb process.
Oh, interesting.
So that is a new research that has just
Wow.
come out like in the last 2 years, but that's quite an extreme, adaptation.
What was the instrumentation you used to look at the vertebrae?
So I used a scanning electron microscope,
There
like
you
most
go.
of these things, but also we tried to CT scan some of this stuff, but the problem is the cytorite is so dense in minerals, such as pyrite, it rebounds all the X-rays. So it just looks like a starburst pattern of, one thing you can do with these guys, a quick way of looking at them is if you have Photoshop, take a good picture of the animal, put it on Photoshop and invert the image.
Oh,
And if you invert the image, it shows you the positive relief of the negative impression.
Oh, excellent. Yeah, because most of us walk around with a 10x jeweler's loop. And
Okay.
I don't think ESCONI has the budget yet to get an electron microscope.
If they did, that'd be great.
So we're a little handicapped in terms of our identifications, but that's magnificent, really magnificent. And I know there was another specimen at the Milwaukee Public Museum that was quite interesting as well.
Yeah. That's definitely a new animal. I got to get my hands on that at some point.
Yeah, there's another one, but it's only a half, and we don't know. The collection was a donation, and a lot of them were single pieces. So
Yeah.
maybe it's in Japan.
Maybe.
Oh my goodness.
That,
What made Charlie do that to sell those? I mean, they're tetrapods or
Very interesting.
vertebrates. Oh well, whatever.
Andrew, it might be kind of bulky to carry an electron microscope into pit 11.
I was curious about how much he may have gotten for those. Was it worth it?
Oh yeah.
Probably not.
Who's supposed to do that?
So Arjun, should we be looking at some older strata for an earlier adaptive radiation or something farther down the tree?
Yeah, so because Mazon Creek already shows a quite diverse like adapted fauna, when the actual adaptive radiation, that explosive radiation of amniotes happened, probably was somewhere in the Mississippian. So a little older than Mazon Creek. But when you look at Mississippian strata, you get things like Embolomiers and Acanthostaga and stuff like that. You don't get very derived things. So currently there's this gap where all of our Mississippian strata are giving these super primitive animals, and then everything in the Pennsylvania is turning up really derived. So you should be
Romer's
looking.
Gap again?
Sorry?
Is that Romer's Gap?
No, Romer's Gap is even earlier, but Romer's Gap is being filled in by people like Lauren Salan, who you guys had here. She's really finding, a lot of fish. They're actually finding fossils within Romer's Gap now.
Yeah, that's what she was talking about.
So we visited a Mississippian site in September up in Michigan. It's the very latest Mississippian, right at the end before it becomes Pennsylvanian. But it's all marine. There's nothing terrestrial there.
Interesting. There's a site in that John Balt looked at that's Mississippian, I think within Illinois.
Yeah, in southern Illinois.
Yeah. That's sort of that's produced a few microsaur-like animals that I've always wanted to look at, but John passed before I could get to it and ask him, unfortunately. So I don't know much about that site.
Yeah, it's a quarry in southern Illinois, and it's been abandoned for years. It's way up. If you look up there, you can actually see where the tetrapods would be. But it is an old quarry. I can't think of the name of the city it's by. It's blanking, but I've been down there quite a few times. But yeah, the actual layer where the tetrapods would come from is not really accessible.
Oh, interesting.
That's by Vienna, isn't it?
Sorry?
John, is it by Vienna, Illinois?
It is. I am trying to think. I think it's east of Vienna, and it would be then a little bit north of there.
Oh, is it Goreville?
Goreville, hey.
Goreville. Yeah.
Yeah.
Okay. Yeah, they call the specimens from the Goreville microsaur.
Yeah.
I don't think it's named or anything like that. Yeah.
I don't know where there's any material, but yeah, that was the site. And the quarry wasn't really much of a quarry, but doesn't have to be if it produces that material.
Yeah. Well, maybe one day we'll get back there too, hopefully.
Well, if you get the land and come in from the top.
Yeah. So actually in Joggins, where I've done some work with my supervisor, some of the localities are very vertical like that. We've actually rappelled down the top. And one of my times rappelling down, the rope snapped on me. And I fell at least 20 feet down to the ground, but managed to survive that. It's got to be done right, but it's possible.
So what is driving this loss of limbs in these animals? It seems like something has to be driving this because it keeps showing up and it's fairly early.
Yeah, I think what's thought in modern reptiles is reptiles have the ability to turn the genes to lose their limbs on and off within the same clade. So for instance, fossorial skinks, when they're burrowing in different types of sediment, so liquidy sediment that is soft versus hard sediment, when they're burrowing in hard sediment, they'll re-evolve limbs. But when they're burrowing in loose sediment, they don't need the limbs, so they actually turn off the genes for the limbs. I think something like that is going on in these guys. They're burrowing in different types of sediment, and based on the use of the limbs, they evolutionarily lose them based on the environments they're living in. So things at Mazon Creek are burrowing in pretty loose sediment, even if they're not living, directly in the Mazon Creek estuarian setting. They're from the near shore deposits near Mazon Creek. And if you look near shore to a delta, you get all this swampland, right? So that's where these things are probably living in. And loose burrowing animals don't really have much need for limbs when they're burrowing. They usually sidewind and use their heads to burrow. They're head first burrowers. So that's the theory about Mazon Creek, that these things are probably just evolutionarily losing it based on the environment they're living in.
Arjan, you showed a marine iguana from the Galapagos.
Yeah.
Early in the presentation, and your description right now makes me think of the flightless cormorants, the cormorant birds, where they have no land predators, an abundance of food, and really no reason to fly or migrate anywhere. So their wings at this point are just vestigial little nubs, and they're mainly swimmers. So again, it's not so much environmental pressure as it's just the sort of lack of other pressures requiring flight or fleeing predators, etc.
Yeah, I guess that if you don't use it, you lose it. That's sort of the deal, right?
Yeah. So they're like caught in this evolutionary, they have these little nubs and they just stand around drying their wings like fingers,
Exactly.
Yep.
Yeah.
I got a picture of a cormorant from Naperville Riverwalk. Believe it or not. Two comments. Number one, have you done anything with the material? I obviously with John Bolt, you must have seen the material from Delta, Iowa.
Oh yes, yeah. So John had a student working on it, and he himself was working on it with God, I forget his name, Eric Lombard, which is another paleontologist out of, I think he's Illinois University. They published a bit on it before John left us, but the other student, his name is Ben O'Toole. He, I believe, is working with Mike Coates at University of Chicago right now, and he's working on that material. So I've seen the material in John's office, and I looked at the specimens, but it's Ben's project, and I guess he'll get to it when he gets to it. Got to leave some tetrapods for everyone to just describe.
Second question is, and this may be not something that you were concerned with because of being in the Carboniferous, but the reduction in digits from 8 to the, I guess, magic number of 5. And I'm not sure, did Tularopon have, was it a 5-digit, I can't remember if it was 5 digits or not. I know
I think it's got 7.
I'm sorry?
I think it's got 7 to
It
learn.
does have 7, okay.
Yeah.
But reason why the magic number of 5 occurred?
I think that's probably fixing on an evolutionary pattern, but the pressures to Y5 are kind of odd because it never really did fixate on 5. If you think about this, amphibians, like frogs only have 4 digits in the front and they have 5 in the back. And then if you think about these animals I've described today, some of them have none and some of them have around 3 to 4. So there's definitely a lot of experimentation within early tetrapods. It fixated on 5 within mammals like us, and same with the mammal relatives synapsids. Synapsids in the fossil record, these things called pelycosaurs like Dimetrodon, all the way to us, they undergo their separate evolutionary history and are pretty canalized in their morphology. I think it's something to do early on in their evolutionary history that does that. But within reptiles, there's a lot more experimentation. If you think about even birds and dinosaurs have reduced it to 3 or something in the front, and around 4 to 3 in the back. So that sort of, within reptiles, there's always been more experimentation on the reptile amphibian side. But within mammals, you have that fixating on 5 digits. Why that is, I don't really know in mammals. But yeah, that's what I'd say to that.
But yet the guinea pigs have like 3 toes on the back and 4 on the front.
Yeah, there's probably some separate loss. But like that's the thing. When we say about fixing on a magic number, there's always exceptions to the rule.
Yeah.
That comes up again and again and again. But generally, I think that even other areas in mammals are very restricted, because if you think about the diaphragm in mammals, that really restricts the vertebral count. One of the features I showed in the microsaurs and fossil reptiles, you have things like snakes getting to over 100 vertebrae, but all mammals really stay short-bodied. Even things like ferrets and like mustelids, they still really retain a fixed number of vertebrae. So within mammalian history, some people argue that the endothermy and warm-bloodedness of their metabolisms keep everything in line with a fixed evolutionary formula, where you can lose things, but it's really hard to gain things again.
Would that apply to birds too then?
Yeah, that probably does. I don't study birds, so I can't really speak too much to that. But yeah, I'm not a dinosaur guy. Although when I was a kid, I probably wanted to be.
Everybody did.
Yeah. All right.
Any other questions for Arjun?
When tadpoles first developed, do they have a correct number of toes, fingers?
Well, when tadpoles undergo metamorphosis, they at first have none, right? They start off as like little worm things. Then they develop limb buds, and the limbs generally have a polarity. They'll develop usually pre-axially or post-axially, so either thumb or pinky dominant, and they appear not all at once. They'll appear like either this way, this way, this way, this way, or they'll appear this way, this way, this way, this way. And that happens depending on the species. So they don't have all the features of the limb appear at once. It's rather a gradual change. Does that answer your question? I'm not sure.
It seems like, except for mammals and possibly birds, that the morphology is fluid throughout the, especially the reptiles and amphibians. It's not, there's no real definite pattern to it.
Yeah, and I think that that's sort of a realization in modern biology that like when paleontology was young and people like Romer were around, they had these ideas based on our living organisms and applied them to the fossil record that, based on the living, that these have to fit in morpho-functional categories. But now, knowing how genetics works and developmental biology and how evolution actually occurs over large timescales, we see that morphology is very plastic in these guys, and that convergence is rampant in the fossil record. You have morphology similar evolving between unrelated clades, even within recumbirostrans, like I showed you guys, that snout morphology evolving more than once in the same related group of organisms, and body elongation and limb loss occurring multiple times in the same group. That's sort of an experimentation between the environment these guys live in and the animals fit towards the environment. They're kind of playing around with what they can do around before they land on a particular morphology that's successful to them.
Any other questions? Anyone?
Yeah, you mentioned once or twice latex peel as part of the research. Is that a common technique? It seems like that would possibly potentially cause damage to the specimen. I was just wondering if that's a common technique or not.
Yeah, so latex peeling for fossils that are negative impressions usually, that's the bone is usually removed or was never there, and you got the impression of the bone, is a technique that Donald Baird coined in the 50s. And it was largely applied to these concretions and to the coal fossils at Linton, Ohio. It does damage the specimens, but it is one of the most high resolution, best ways to study these fossils. Now we're experimenting with things like CT data, which is why the last few organisms you saw in my talk, we didn't peel. We didn't peel because the museums didn't want it, because they didn't want us to destroy the specimens, and we didn't want to destroy the specimens. So we CT scanned a few, and generating a surface model, you can actually take the image of the CT scan and invert it to make a digital latex peel. And then from that, you can 3D print it and have an actual latex peel without having to peel the animal, right? And that's sort of the progression of technology and how we integrated in studying these animals now. But I'll say this. I recently latex peeled with another researcher a few animals from Linton, and we stuck the scales that were in the latex peel under the scanning electron microscopes. We saw details in the scales, these little nubs and ridges that the CT scan could not pick up. So there's something to be said about keeping some old techniques in areas to study the anatomy as best as possible, and then when possible implementing these new techniques. Newer is not always better sometimes.
Interesting, thanks.
Yeah, no worries.
So are there any other sites around here in Illinois and in the general area of Mazon that would be useful to sample?
You know, within Mazon Creek, what I came to found is that it's not really one locality, right? There's all these pits. I think what would be useful to kind of look for is new outcrops of the Francis Creek shale in different areas. One of the animals that was described, the Diablo rotor, is actually from Fulton County. That's quite far off from the stuff in Pit 11. So searching places like Fulton County a little more, and it was donated by a man named Leonard A. Lasco. I don't know if anyone knows who that is, but searching through the notes and old history of this area, notes I got from Donald Baird, it appeared Leonard A. Lasco was a private collector that Donald was in contact with regularly, and donated a few specimens to the Augustana College at Frixell. So I think searching Leonard's sites at Mr. Lasco's sites near the Fulton County area for more animals of this would be better. Sorry?
There's a coal mine, Astoria,
Yeah.
is in Fulton County. That's probably where that came from.
Yeah.
Interesting.
It's supposedly on farmer's property. And it's just sitting there from what I understood or understand.
Interesting.
Now, might be the same area, but if memory serves, and you guys would know more about it with Mazon Creek, but weren't there some areas over by Peoria, in the area of Peoria? Or maybe that is
between Peoria and Macomb. And I've heard of things around Macomb too.
Okay.
Isn't Astoria also where almost every concretion contains a Belatelsin shrimp?
A shrimp, yeah.
Yeah.
Yeah,
Nearly every.
that's the sunspot mine.
Right.
Hey, Bruce.
Yeah, Rich Rock was the guy that collected there extensively. And that is the spot where 90% of the oval concretions were a Belatelsin shrimp.
Also, that site, I've been told, based on what was found there, may be more terrestrial in origin, because you seem to get less of the fishy things and more of the kind of proximal lungfish to tetrapod animals. Yeah, that might be an area worth looking at.
Arjun, if you bring up Google Maps and go to Astoria, Illinois in Fulton County, zoom in and you can find the characteristic sort of landscape marks that were surface mining and stuff like that. So you could see the strip mine type stuff you see around Braidwood and Wilmington. Couple sites right there. It's pretty obvious.
Oh, yeah. Big satellite. Oh, yeah.
I know.
Interesting.
On satellite maps.
Now, way back in the day around Pontiac, Illinois, which is known for the crinoids, there were some tetrapod bones found in a couple of the quarries there.
Interesting.
And I think there's one that was a student found and gave to Illinois State University in Bloomington Normal. That's in Normal.
John,
Yeah.
was that in the Black Shale?
No.
Oh,
Believe it or not, this was in limestone.
Oh my goodness.
There was the one that was found at Lone Star.
Oh, I've got one of those.
One of the tetrapod bones?
Oh yeah.
Okay.
Not the big one. I got one about this size, probably an amphibian.
Interesting. You should send me pictures.
Okay.
Yeah, I'd love to see them. Is the thing, like, I feel like my gut from looking at the Mazon Creek fauna and the stuff I've seen is that there's a lot more diversity than has been previously recognized. So some of the things that people are like calling amphibians are probably entirely different species. And that'd be really cool to sort of get in the literature. Even limb bones can tell you a lot. The difference between a reptile and amphibian limb bone are like night and day. Yeah.
Well, that site that I just mentioned where the tetrapod limb bone was from, called Lone Star, that's in the process of being turned into a fossil park. So if the
wrong side of the river,
if the Illinois budget doesn't crush it in the meantime, then maybe a year or two down the road we'll have some access to that.
Huh.
Dave, there's always a chance, but the two or three that I know were found on the other side, the south side of the river, in the south part of Lone Star.
Yeah, I don't have a good feeling about that
No.
area.
Not at all, since those people were killed down the river there.
Yeah, I don't think that's gonna survive.
Nope.
Are there any areas in Mazon Creek that would benefit from like a full like field party excavation? Because I asked this because while I'm transitioning to a role at Harvard, they have a lot of money and they have a grant for field work. And I'm faced with the option, and it's quite a big fieldwork grant, but whether or not I want to take this money to go back to Linton, Ohio, or Mazon Creek. So if there's localities in Illinois, and particularly Mazon Creek, that you find are pretty productive and would benefit from getting something like an extractor or some actual big equipment and some rock saws and hire a field team, that'd be something to let me know as a group.
If you guys remember, that was always Andy Hayes' idea to go there and essentially disc the area and bring up, get rid of the vegetation and bring up new material. That went by the wayside.
I think if you put a trawler in Braidwood Lake, you could probably dredge up concretions by the tens of thousands.
Wow.
I mean, they're sitting in silt at the bottom of the lake. Pit 11 is mostly submerged now.
That might be
even if you dredge the shallows just near, but I don't know what kind of permission you need to get to do that. But even if you just dragged the shallows around the shoreline, you would come up with thousands of concretions. So,
and a lot of unhappy fishermen.
Yeah, let's rent a shrimp boat and head out to Braidwood Lake.
I know of only one legless amphibian. It was found on the islands in Braidwood Lake.
And where did it go?
Rob still has it.
Who has it?
Rob Coleman?
Yeah. Yeah, I was there, of course. I got to follow in front, if that makes sense.
Need a tackle.
So if folks know of some site that, especially something that's going to be privately owned, like everything else here, if you've got some site in mind that you think might have the possibility, we can let me know where it is and go ahead and do the initial contact on our behalf. We have sort of a package of documentation of insurance and the state law that limits liability for landowners and that sort of thing. We can try. All we can do is try if we know who to talk to and where to look.
Okay. The other, do you guys have any relation with Tom Testa as well?
Yeah.
Oh yeah.
Yeah. So Tom, I met him a few years ago. I don't know if he's on the chat right now, but he donated a bunch of his stuff to the Field Museum, and I recently looked through his stuff again and pulled out one specimen that is a new tetrapod from there. I had originally kind of glanced over and looked at it, and this thing is a beautiful legless amphibian like Phlegethontia. I was thinking of naming the guy after him. So if anyone has his email or how to get in touch with him.
He's on Facebook if you're on Facebook.
Oh yeah, I don't have him on Facebook. I'll look for him then. But yeah, I wanted to run it by him if I could name the species after him. I thought that'd be a very fitting reward for him.
Yeah.
He's got an insect named after him.
Oh, interesting. Cool.
Yeah. Jack Wittry is in contact with Tom.
Ah, okay. Okay.
So that might be another avenue.
Do you have your email?
Yeah. My email right now is Arjan, ARJAN, .Mann, MANN, at Carleton, CARLETON.ca. It will change once I get stateside to Harvard and they activate that email. Right now I'm being paid by contract because of the pandemic. Whatever, I don't know honestly how long that's going to take, but hopefully sooner rather than later.
Harvard.
Yeah.
My daughter went to Cornell, and every time anything in Harvard comes up, it's the old saying that all the Cornellians had: Harvard sucks. Harvard sucks.
Yeah, it's
an Ivy League thing, guys.
Yeah, even between some of my colleagues that they all have scoff at Harvard sometimes, but they have a good program going on right there with Dr. Stephanie Pierce, who will be my supervisor. She's supervising a lot of biomechanist people. But what I hope to do there is to bring a systematic component back to the department and start really describing a lot of new species. But as I said, they have this thing called the Putnam Grant, which is a fieldwork grant to launch a full expedition. And from what I hear in the department is that they don't have very many systematists, and the grant is designed for systematic research.
Yeah.
So it's basically a shoo-in for me.
This is happening all over. Systematics is not at the top of the list anymore.
Yeah.
Even I was talking to Dennis Collada about it. I was talking to Roy Plotnick and out at PRI, Warren Ullman. One of my essays in PRI was about that, about the lack of systematists and the detriment. And apparently it's not just paleontology. This is actually happening in all the natural sciences, biology too,
Yeah.
which is sad.
One thing you mentioned, Roy Plotnick.
We all do.
So I just saw the other day, they're posting a postdoc position to transition to a 10-year faculty member at the University of Illinois, where Roy Plotnick is. I was thinking of applying for that. If you put in a good word for me as a group, I'd appreciate that, because getting a position in Chicago, whether it's at the Field Museum or anywhere in the area, would really foster this relationship, and I'd be boots on the ground at Mazon Creek and really be able to power through this research. Harvard's a while away, but that's a temporary position for me as a postdoc. If I could get a faculty position in Chicago, that's my dream.
Well, just really contact Roy. Is very approachable.
Yeah. All right.
He is really one of the great guys, and he's just retired from the University of Illinois Chicago.
Oh wow. Okay, I saw him listed on the faculty page, but I assume they're doing a general earth science call out. Basically, they want to hire an earth science position, but they don't care whether it's a paleontologist or a geologist. But I assume he'll be on the review committee for whoever they hire. Yeah, I'm hoping that'd be a great position for me.
He is a very approachable person. In fact, I think he was just co-author of a paper on a Mazon Creek arthropod or something.
Oh, cool.
Guys, help me with this. Chirp, chirp.
Oh, no.
No one's reading the literature.
Last one I saw was on the jellyfish not being jellyfish.
Or something like that. I mean, it's Mazon Creek. Come on, it's
Yeah, but that was never even published.
Yeah, I think that was published.
No, I've got something on it.
It probably was a poster session or something like that.
No, go to the JP. I belong to it, so I can go in there and get the pre-articles, and I'm sure I saw one with Roy in there. I can go to and check it out, but obviously not right now. That would be too cumbersome. But I'm sure I remember him being
He's on something on Essexella from there.
Yeah,
that's the jellyfish.
Yeah.
Yeah, maybe he got it published.
So what's Essexella now? Is it a jellyfish or no?
Oh, that's very controversial.
Okay. The invertebrate side is not as well known to me, but I'll let you guys know something. Actually, I started at Mazon Creek working on Belatelsin in my undergrad under a professor, Mark Laflamme, who's an Ediacaran paleontologist at U of T. And this stuff was all Kathleen Ramsdale collection at the Royal Ontario Museum that Jean Bernard is curator of, who will give you a talk later. I found that Belatelsin majester is definitely two species at least. And there's one animal, there's basically an animal I was going to designate a holotype, but then I went off to work on fish and tetrapods and never got to it. So I always think I got to name that at some point, but there should be at least one more species of that guy there.
I had a nice one open up about a year ago.
Oh, wow. The shrimp are just so beautifully preserved from that site. I was really shocked by, they're preserved better than the tetrapods, which is ironic, right?
A lot more common too.
Yeah.
That picture that you showed of the people collecting on a kind of a reddish colored bank.
Yeah.
That I think is one of the sites that we go to a couple of times a year. I think that's
Looked like Braceville.
Braceville, yeah.
Yeah, that's Braceville.
That was a site I was thinking that could benefit from like mass collecting. I don't know what the rules are there.
Well, that's spoil pile from a shaft mine,
Okay.
and the owners of the site use it for fill material. So we get in there a couple of times a year for 2 days each. So we're in there 4 days out of the year collecting and digging and that sort of thing. And I don't know if they would really want a big change made to that hill.
Okay.
Further, it's largely marine species, clams, polychaetes, jellies. So I don't know what the frequency of tetrapods would be if it's not zero.
Okay.
If that's the aim,
There's Joan.
Hi, Joan.
There's Joan amongst the trees.
I know, Joan.
It's a background of it's a picture I took in California at the Calaveras Big Tree State Park about 2 years ago.
Huh.
The perspective looks like you're rappelling,
Let me
like
leave
face
it
down,
at-
like a la Mission Impossible style.
I laid my camera on the ground, pointing straight up,
Beautiful.
took a picture.
And Arjun, Jack Wittry said a couple of months ago that he's starting on a new revision to the fauna book. So I think he'll have some changes to make.
Oh, if he wants to get in contact with me too about that stuff. I've never met Jack Wittry.
I think he's on the call here.
Is he?
If it's him that he's muted.
Yeah.
Him Jack down there.
There is a Jack on the call.
Oh, well, I'd love to get in contact with him. But yeah, I think that would be in need of a revision. There's just so much stuff coming out of just the new taxa we're finding there, not just at Mazon Creek too, but a lot of my postdoc is supposed to be at this site, Linton, Ohio. And I remember a few years ago, I had a trip to the Carnegie Museum, and a collector named Scott McKenzie, I don't know if anyone's familiar with him, but he donated a bunch of material, and piece after piece was new animal. Me and the curators sat beside each other, and we were finding big synapsids, we were finding herbivorous animals, and that's one big thing too. It looks like the origin of herbivory occurs actually in the Carboniferous. So that rewrites the textbooks, because a lot of people are thinking the origin of terrestrial ecosystems and the modern structure of the trophic web really occurred towards the Permian and Triassic. Finding these animals as early as the Carboniferous really means that after the adaptive radiation of amniotes, however early that was, you had a massive explosion and most diversity was filled, at least at some level. And that stability of that ecosystem level has maintained all the way through today, through all the mass extinctions. And that's sort of one big aspect of my research. So there's a lot to be described, a lot of new stuff to be described. And that's the importance of systematics. That's what
Exactly,
systematics tell you.
Yes. That article by Roy Plotnick, I looked it up. I was mistaken on that. You're right. The one on Mazon Creek, he hasn't published yet, but this was on a Silurian phylocarid. So sorry about that. It was a mistake. I'm getting old. Well, wait a minute.
Looks
I am old.
John, it did look like he wrote a paper. It was 2003 though. On
Yeah, that could be. But this is this one on the Silurian phylocarid was in the volume or issue 5 of this year's JP.
Arjun, have you ever heard of an animal that's that's typically referred to as a blade, a larval fish, or potentially, I think some people have said larval amphibian. Do you have any?
Yeah,
opinions about
I
that?
actually have. You know, I'll tell you guys something. I do a lot of like surfing on the internet for Mazon fossils sometimes. I swear to God, if you on Google, if you type in larval amphibianus, this picture of a beautiful amphibian larvae from Mazon Creek comes up. I don't know who owns that. I tried to track down, on the fossil forum, who might have owned it. I sent emails out, got nothing back. But that collection of fossils that are blades, amphibian larvae, fish larvae, they offer a world of developmental information on early tetrapods. That is so unique that you can get at Mazon Creek the earliest developmental stages of tetrapods and compare these things one-to-one with embryos that we grow in a lab. So if anyone has an idea where any of those specimens are, it would be the world to know where they're found.
Blades are something that aren't that uncommon and are still found in Pit 11.
Interesting. I haven't seen very many of them, but I'd love to see pictures.
And the genus is named after our club, Esconichthys.
Esconichthys, yeah.
Oh, so Esconichthys, that was one of the things. That is a project that a colleague of mine and me are supposed to be working on. What he thinks Esconichthys is a collection of rhizodont larvae, lungfish larvae, and tetrapod larvae. So he's separated them out into various morphs. Some of the Esconichthys, where the fins occur closer to the head, are generally characteristic of rhizodont. And some of them bear actually rudimentary pectoral elements, where some of them bear very tetrapod-like positions of the limbs. So that's kind of an interesting project that he's contacted me about working on. But the methods he wants to go about by analyzing this is looking at specimens over large museum collections and sort of doing a statistical landmark method and binning them out by morphotype. It's a big project. I don't know if we'll see it in the next 10 years because there's a lot of material to siphon through, and some of these things are so squishy looking.
Tell him he has to leave the genus name alone.
Yeah. Well, we can always erect an Esconichthys-like name for a tetrapod, Esconerpeton, if you guys, let me know. If you guys have a tetrapod you want to donate
What's that?
to the cause.
John, what have you got?
That is that's the tetrapod limb right there.
Pull it back, pull it back, pull it back.
I forgot that I had it here upstairs.
And where's that? That's not Mazon, is it?
Oh, no. This is Pennsylvania, probably bond formation. So it's upper Carboniferous. But that's the small. There was a large one found. I thought there might have been a third one found too. But
So
this is the one. That's the one that I have.
That looks like an embalomere. Embalomeres are animals like Spondylerpeton from Mazon Creek, if anybody's familiar with that, or Anthracosaurus russelli. Those are pretty interesting, important animals. And that's Mississippian, you say?
Pennsylvania.
Oh, Pennsylvania. Okay.
Yeah, upper Carboniferous.
Yeah, that'd be great. I'd love to see that site at some point. That's pretty good, though.
Pardon me?
That's pretty good. Pretty good looking.
Yeah, I was surprised. I'm not a vertebrate collector. I don't, I never really collect many vertebrates, but except for shark's teeth. But unfortunately, there's in this Lone Star, there's two parts to the Lone Star property on either side of the Vermilion River, and unfortunately, all the ones that I know of, the tetrapod bones that were found on the south side, and that land is. You step 1 foot in there, and you'll probably be thrown in jail. Yeah, they're very touchy about that part because somebody was going down the story I heard. Some people were canoeing down or boating down the river, and something happened, and one or two of them died. Even though that's technically state property because it's in a navigable stream, but the company's upset about it. The north side is right. We're trying to get that into a fossil park. And that side is notable for a lot, well, billions of invertebrates, literally billions of invertebrates. And for the Peru monocline, which you can see there. In fact, you're actually collecting up on the, as you walk strike, you're walking up the monocline, which is kind of cool.
Shark teeth.
And yes, at one time, there were a lot of shark teeth there in the Modesto, I think it's the Modesto cyclothem, the one on top of the Bond Formation. It was in a light gray shale. There were several times that Tom Williams and I were there, that we each came home with 400 shark teeth.
Oh, wow.
Yeah, but shark teeth. I mean, come on, you're not there.
Can I share my screen with you guys again?
Sure.
You guys have kind of got me interested in. I want to show you a tetrapod that I've found. So, can you see this animal?
Oh yeah.
That's a tetrapod?
This is one of the blades or larvae that is clearly a tetrapod larvae, not Esconichthys.
Really?
So if we zoom in here,
You can see its skull.
that's the skull. This thing here is a limbud. That's the ulna, radius. You got the digits here. This is the skull. Obviously, we have teeth, tetrapod-like configuration of the snout. And if you look at the back here, you can see the tail region clearly defined, and the vertebrae of the tail and hamal spines.
Mm-hmm. Wow.
No, that's cool.
Yeah, and the whole thing, instead of kaolinite, is this arsenopyrite.
Oh, yeah, be careful.
Arseno?
Well, it's not really arsenopyrite, but it's some sort of
It's supposedly arsenic, but
yeah.
Believe it or not, we had that at South High when I was there teaching, and we kept it in plastic bags. The students never touched it, but that's true, pure arsenopyrite.
Yeah. So
that is so cool, though.
This animal is going to be super important. It's one of the specimens that we singled out to work on, that we want to submit this guy to something like Nature, because this is the earliest stem tetrapod embryo, which it provides a polarity for developmental traits within all of tetrapods. What is interesting about this guy is that at smallest level, the anatomy is pretty similar to at the largest level. Nothing changes. So this is something called like a direct developer. There's no metamorphosis, no gills, no eggs, no yolk sac. So it's developing at its smallest stage, just growing larger till the animal looks like the way it is. So basically what this shows is that early tetrapods, like fish-like stem tetrapods, didn't undergo an amphibian-like metamorphosis.
Now, this is really a stretch. That couldn't be a live birth, could it?
It probably was. Because it's so small and looks the way it does,
wow,
tiny unossified level, it probably was what you would consider a water live birth in the water.
If memory serves, I can't pronounce his last name. Per Albert, or
Per, yeah.
He, I think they found the first Devonian tetrapod that gave live birth, if memory serves.
I think so. I don't think that's actually, I don't know if I know of that study. I don't think that might be a thing. I'm not sure.
We'll have to go back and look at our worms and our blades then.
Yeah.
Yeah, there's certainly diversity in there.
What's the size of that?
That's about, let me check.
1.5 inches.
Yeah, about an inch. About an inch, less than an inch almost.
Okay.
Some things we call worms might actually be something else then.
They could be. You got to look carefully. I'm trying to find if there's I'm still sharing my screen, right?
Yes.
Yes, you are.
Okay. Try to.
Some of those might also be thought, some of our leeches might be not leeches.
You can
Oh,
see
look at that.
the underside here. You see the two jaws, the maxilla, the eye.
I'll be.
These are the ribs.
Is that a limb? Yeah, right
That
there.
would be where the limb is.
Okay.
Not very well ossified. It's much better represented on the other side.
Yeah.
But the tail's pretty telling of an embalomere. See if I have some better pics. Let me, while I got you guys here, let me also try to find the other Milwaukee Public Museum specimen. Milwaukee.
Arjun, actually, me, Andrew, and Bruce on the call actually were up there helping them identify some things about, probably, was that, Andrew, 4 years ago?
Yeah, we started on the project, about 4 years ago. I think a grad student was helping Pat Burke sort things out from this massive donation that was completely unidentified. And it's still in progress, but by all means, when we pulled those amphibians out of the lot, it was quite exciting.
Mhm.
There were some nice fish too, and I think a shark was in the group.
Yeah.
I don't know what I called the image. New microscore, maybe?
Remember what it looks like? Oh, yeah.
Any of you have good photos of it.
Yeah, Arjan, I've got pictures. And there's also Bruce and Rich, if you remember, do you remember the large concretion that had what looked like just a skull in the center of it?
Yeah.
It was decapitated, detached from its body. But looking at Arjan's images, I'm feeling like that might have been an amphibian skull.
Mm-hmm.
There's also that weird leg or.
Oh, that's not it.
Right, that huge crustacean leg. I mean, it was like a Red Lobster snow crab week.
That's one way to say it.
No, I mean, stretched out, it was like 3 inches long.
Mm.
That's pretty big for a crab leg.
There's a better image of that, this guy's leg.
Wow, that's impressive. Now I want to reevaluate my collection.
Problem is I got too many projects on the go.
Well, now the next time we go to the warehouse, guys,
Yeah, I know.
we're gonna have a whole different viewpoint on the worms.
Arjan, I don't know if David sent you a picture of the singular skull from Milwaukee, but I have an image of it, and I'll forward
Yeah,
it to you.
I don't think I've seen it. So
Okay, I'll definitely after your talk. I mean, I'm now very intrigued by it. I'm no expert on the matter, but it was definitely intriguing.
okay, yeah. Well, sorry, I couldn't find it.
Wow,
But
that's incredible. Oh, so's that.
all right.
Sorry, I'm not sitting at my computer, so I don't have access to those images right now. So Arjan, I'll send them to you on your email. I'll send
All right.
things of those. Yeah.
That sounds good.
Yep.
If you think we know somebody or can help talk to somebody, let us know.
Okay. So I just don't know what I did with that photo. But yeah, no, anyone wants to ask me any more questions, please.
Oh, Arjan.
Yes.
Andrew.
Andrew.
What's your viewpoint on the Tully monster?
Oh, okay, yeah. So whether it's a vertebrate or not.
I mean, since we're talking vertebrates all night long.
Yeah, I feel like looking at its anatomy, it really does not look like a fish. I know there was that study that came out that the eyes have pigment that fish have. And then there was one that the body plan, the notochord, looks like a fish. Then there was a paper responding to it by all the fish workers, Lauren Salan included, that said it wasn't a fish.
Right.
When all the fish workers get together and say it's not a fish, that's kind of telling. You know, it's like, what, those are the experts. And it wasn't just one of them. It was like 6 of them, right? I think the evidence probably is a little inconclusive. One issue that I've heard with the methods using to identify the soft tissues that they were using were done by this researcher, Yasmina Wyman, she's a German researcher at Yale right now. Her methods have recently been criticized as possibly experimental error. And if they are error, it makes all those results null and void. So I'd be careful about buying, because it's such, and it's nothing bad on Yasmina. It's she's doing something so experimental, you often fall before you start running, right? So I'm sure she'll work it out eventually, but those results are really tentative. Expect at least 3 more times it to change position in the next 100 years, because until you find a fossil that actually preserves a goddamn vertebrae, you can't really say it's a vertebrate.
Well, I mean, and we're a group that uses a jeweler's loop to make our conclusions for the most part. Yeah. But I think there's a silent majority among us who believe in this sort of shellless mollusk.
I'm not silent about it.
The not so silent majority.
I'm not. I didn't want to bring it up. I'm glad somebody else did, but there is absolutely no question that Tully monster is a shellless mollusk. Period.
And Essexella is not an anemone. I said it. No way.
It's good to get that out.
Yeah, I know. I had to get off my chest.
Vertebrate, my.
Yeah, I'm not. What did Lauren Salan say 2 weeks ago about it? I'm curious to hear her.
Oh, we didn't listen.
I can't remember.
I believe.
Oh, I wasn't sure. Interesting.
Actually, you do have her presentation recorded, so
we do. It's on our website too. You could download it and watch it, or watch it on YouTube.
Okay.
But I think she said it wasn't a vertebrate, if I remember right.
Yeah.
A mollusk.
Yeah.
If you don't mind, I got my one and only blade and took a picture of it. Do you think I can share it?
Yeah, absolutely.
Make it so.
I'll send you a couple pictures, Arjun. I've got a couple.
Do.
Not great.
Ooh. It's a little dark, but
I like the pigmentation in it.
Yeah.
That's interesting.
I'd have to see with photographing these things, a real trick I found is that low angle light.
Yeah.
Really lights up skeletal elements. But this one looks a little bit more like a wormy thing to me. What's the hagfish from Mazon Creek?
Don't know.
Oh, there's a lamprey.
Yeah, lamprey. This may be one of those. Kind of reminds me of it vaguely.
So you're back to a Tully monster.
Mhm.
Yeah. I didn't see any tetrapod characters there.
I didn't see any either.
Yeah.
Wishful thinking.
But yeah, you never know.
Yeah, that's right.
I have a nice blade with eyes.
Yeah.
So I take it the spiny fish is a spiny fish.
Yeah.
All right. Anyone else have any questions, or do you have any other good pictures to show anybody?
I mean, I might be able to go grab the blade.
Yeah, tell me you're gonna get the blade.
If you hang on 1 second, I might be able to grab my blade and just hold it up to the camera. Hold
Please
on.
do.
Hold on. Now that we're doing this,
There's quite a lot of material in our warehouse, Arjun, that came from Pit 14, which is a little bit different area. So, you know, we'll look at that next time we go to the warehouse.
Okay. Any notable tetrapods or nothing off the top of your head?
I do not know.
Okay.
But there's,
Yeah, this is over.
Dave,
I know
there's,
that
there's
I
so
can
much
tell you
that we
there's-
haven't gone through though.
Yeah, there's a lot of stuff we haven't gone through. I know I brought home a drawer full of Iniopterygian material
Okay.
from in black shale.
That's not bad.
Here's a couple blades. Let me see what you think of these. So here's one that opened up this summer. Can you guys see it?
Oh,
Yeah.
okay. So this one looks like a typical lungfish larvae. This is what might be a juvenile of something like Concopoma from Mazon Creek. Its skull and like sort of fluke-like tail without the big four-limb flippers up front kind of indicate that. That's actually not a bad one. That's a pretty good one.
It's really
I'll send you a picture. I'll try and take a better one. Here's
You know
one
what, guys? I actually found my picture of the Mazon Creek Milwaukee specimen. If you let me share that for a sec.
Sure.
That was bugging me not being able to.
Here, let me stop sharing. This is the other one that I have. Let me find where. How do I stop sharing?
Oh, that one.
Good.
That one looks lungfishy as well, but that's definitely a vertebrate.
How do I stop sharing?
I think you
Try mousing over the top.
There's no like way to stop.
Can't find it?
I can't find a good one.
Can you get to the title bar of the
Oh, I see it on
program?
the right. Stop share. I see it now.
No, I don't know.
Ah.
They may have needed a larger cabinet.
Oh.
All right, let me share my screen real quick.
Um,
So this is what I got a picture of.
Yep.
Is this
That's
the one?
familiar. That, Rich and Bruce, that's the one we saw up in Milwaukee.
I'm trying to stop sharing. Are you sharing something right now, Andrew?
He's doing it, Rich.
He's back. Okay, cool.
And do you see this one? So this is a quick inversion of the image.
Wow.
Arjan, I took a photo of it in really interesting raking light, you know, so like a low angle light, and a lot of the spine elements really popped out.
Please send that material, that specimen photo to me. So I think this is a different form from Diablo rotor. It's probably its own genus and species. It's definitely a microsaur too.
The skull is really defined. It's
Yeah.
it's not dissembled, you know.
Yeah.
And I think they're hind limbs as well.
Yeah, that should be that there.
Yeah.
But
Is
this
that
is
a forearm up there?
really,
Oh, oops.
that's a really great looking animal. One thing I kind of noticed on this animal is that if you look at the tail here, the hind vertebrae, these vertebrae looks like it's been cut off, autotomized, and the tail has started to regrow.
Oh my goodness.
Oh, regeneration.
Super odd thing. Trying to see if the inverted image shows it a little better. Yeah, so this little cone at the end, you can see here, a really small vertebrae after the big ones.
Wow.
That's a sign of amphibian-like regeneration. The
Yeah,
tail.
I mean, and don't skinks like can detach their tails if they're trying to evade prey?
They can, but they usually don't regrow the individual vertebrae. They
Okay.
regrow a cartilage cone, and then the tail on top. So this is kind of odd. This is something that really needs some further investigation, but that's a very unique feature in this specimen.
And there's ribs too. Wow. Yeah, that's awesome.
And 10 o'clock.
All right. All right. That's all I had for you guys right now.
You're gonna have to share the screen, Andrew, and unmute.
He's got his mute on.
There you go.
Okay, I'm unmuting,
There you go.
and then I'm going to hold this.
You have to share, but I'm not.
Where do I
Yeah,
share?
he is.
Right there.
Back up.
He's got it. Back up. Back up. Keep going back.
Clock.
Oh, you might as well close it.
Say something, Andrew.
Okay, so can you see the, it's a dorsal view of blade. You can see the two eyes.
Yeah.
Yes. So that one, because it's got those big fins, has a chance of being a rhizodont larvae.
And that's what you're calling the lungfish, is that it?
No, so the rhizodonts are different from lungfish.
Oh,
it would be its own particular class. That's a unique specimen. That's something I'd like to look at.
very nice. Thank you. Okay,
Yeah.
I'll take a good shot of it and send that in the group.
Perfect.
Now, where do I unshare screen?
No,
You're not.
It didn't go.
You,
you're not sharing.
Okay,
you
it's
were,
good. I wasn't sharing. I don't share.
doesn't play well with others.
No. I'm a misanthrope. And that's not the good blade. I couldn't find the really good blade. That was the second string.
So how frequent do you find these blades?
Arjan, I was searching for my better blade. That's also a dorsal view, and it shows the pectoral fins very clearly. I found it last year in Pit 11.
Interesting. Okay.
At Rich Holmes' secret spot. You'll have to ask him if we can take you out there next time you're in town.
Oh boy.
Mine opened up this summer too, and it came from Pit 11 also.
Yeah,
Was that one that was originally in front of me?
that was Marie's actually, and Rich pulled a pirate act.
Not on that one. That
Oh,
one.
that was a shrimp.
Oh, that was the shrimp. I'm sorry. Yeah. No, so Pit 11 is still productive.
Yeah.
It's just overgrown and it's kind of hard to get in there, you know. Keith has had more experience on the islands, which have produced some interesting things. But we have found blades and tully monsters in areas that you can get into by walking.
Interesting.
Yeah.
That's not bad. That would be something I'd like to see.
Yep.
We do freeze thaw 'em, so as you're probably fully aware that you don't get immediate gratification. It takes
No. Yeah.
still time.
So the really nice blade specimen I found was the very last stone in a haul of about 75 pounds of rock that I took out of Pit 11 last year. And all the remaining ones were jellies or coprolites or non-fossils, trace fossils. So it was the very last one. So it was like one blade in 75 pounds, which might be several hundred rocks. So that's pretty infrequent.
All right. Well, it's getting late here, folks.
Yeah.
Yes.
Any final questions? Final questions. Now's your chance.
Arjan, thank you very much for your excellent
Thank
presentation.
you. It's excellent.
Yeah, and all the
Thank
time
you.
you gave us following. This has been exceptional, really, really nice. And I look forward to being in contact with you.
Yeah, that's exactly what I hoped, what I wanted. You guys are essential to my research because I'm floating around Mazon Creek and doing all this work on tetrapods. I don't know if any of those tetrapod concretions in the past that I've studied would have gotten into museum hands without groups like ESCONI working Mazon Creek tirelessly. And I'd really like over the next few years to become integrated with you guys and be able to give you talks in person, hopefully get down there and meet you all. So This was really great for me too. Thanks a lot.
Thank you.
Thank you.
very much.
Thank you, Arjan.
Good night.
When you're in town, if you do get to come in, let us know.
Alrighty.
Yeah, there are some crazies that still go out to the at 11 and Ponderosa Lake area.
You know, I do have, excuse me, just one quick question. Arjan, I'm wondering, how did you get interested in Mazon Creek fossils to begin with?
That's
A lot of us, you know, coming through the club and, you know, live in Illinois. And so how did you get interested in it?
Yeah, so I'll tell you my life story real quick.
Okay.
My
When I
mother
was 3.
Yeah, my mother worked at the Toronto Royal Ontario Museum when I was a kid. She did a degree in paleoanthropology. So I spent my younger years around the Royal Ontario Museum bugging the curators. And eventually I made my way into undergrad and grad school and was still bugging the curators, got in the collection. And one day, looking through the invertebrate collection, I stumbled upon the Ramsdale collection. And I proposed doing a project on the shrimp. And then I started pouring through all the Mazon Creek collection they had there and found a few tetrapods. And I looked at those things and I was like, I always had them in the back of my mind. When they offered me a master's project, they offered it on fish. If you look at my first publication on Google Scholar, it's on a large sarcopterygian fish. But I kept those Mazon Creek fossil images in the back of my head. And when it came time to doing a PhD, I was proposed a project and I said, screw that. I want to work on Mazon Creek amphibians. And I showed her Hillary all the data I had and sort of formulated my own project based on that. But it was my years ruffling through the collections of the Royal Ontario Museum where I really saw Mazon Creek, saw how kind of underworked the tetrapods were, and really just fell in love with the material. And I think that there's so much opportunity at Mazon Creek to do large scale projects in the Carboniferous. Nowhere in the world do you have an abundance of such a complete Carboniferous ecosystem preserved, where you have things from primary producers, plants, all the way to tetrapods. And that's what makes Mazon Creek unique. And that's what really pulls me to it. It's like a window into the past for me.
Great story.
Thank you.
Yeah.
Thank you.
Yeah.
Welcome to the insanity.
Yeah.
Yeah. Hopefully we get more insane over the years.
I look forward to it.
Just look at us old guys. Yeah.
I know. I was referring to you, John.
Well, you know, I can't help but think about too, when you're telling your story, how important the museum collections are.
Yes.
It's just vital, right? I volunteer at the museum and I'm always thinking about that.
Oh, do you volunteer at the Field Museum?
Well, no, at the Peggy Notobart Nature Museum.
Oh, cool.
But there's in fact, that's where I met Andrew and Joan for the first time. They have a nice size collection there.
Cool.
Yeah.
Which I've been cataloging with Andrew's help for the last 11 years now.
Rich works at the field.
Yeah.
They don't have any tetrapods
Maria and I
unless we revisit the blades.
Yeah.
Well, everybody stay safe.
Yep.
Yeah.
All right.
Yep.
Thanks again, Arjun.
Okay.
No
Yeah,
worries.
thank you very much.
Night.
All right, I'm going to sign off now.
Okay.
Thank you, Arjan.
All right. Bye. Thank you for having me.
Thank you, Arjan. All right. Yeah. Who's going to say?
There's Mark. Hi, Mark.
Hi, Dave. I got some good news for you, but we'll talk another time.
Hi, Deborah.